So in this video, first we are going to see about the main pathway, that is heme synthesis. This is our first video, and in this video we can see that formation of delta amino levulinic acid. So this is the main important starting uh, substrate which is formed in the heme synthesis. First one you can see glycine. So glycine is the amino acid which combines with the succinyl CoA. So succinyl CoA, which is also present in Krebs cycle. So from that Krebs cycle, succinyl CoA can be used here. Combining with glycine, it forms the delta amino levulinic acid using the enzyme that is ALA synthase. So ALA synthase is an important enzyme. Okay. So why this ALA synthase is an important enzyme? Because here this heme, which is formed as an end product, has a negative feedback mechanism for this ALA synthase enzyme. So when heme is more in amount, it inhibits that ALA synthase, and so the formation of delta amino levulinic acid is reduced. Okay. Now the second step, which is delta amino levulinic acid, is converted to porphobilinogen. So porphobilinogen, here this enzyme is ALA dehydratase. So this ALA dehydratase plays an important role. Okay, you have to remember this enzyme which converts ALA to porphobilinogen. So I have kept a mnemonic for the pathways mainly by using the starting words, that is PHUC and PP. Okay, so you have to remember by the for the normal 12th standard, previously they used to write exam known as PUC previously. So I have kept a person with the name PP. So Mr. PP, you have not even completed PUC. Okay, so this is the mnemonic. So PUC, you have not completed PUC, Mr. PP. So here you have to remember an extra one, that is hydromethylbilin. This hydromethylbilin you have to add in between that porphobilinogen and uroporphyrinogen here. This is because this plays an important role in one disease which is known as acute intermittent porphyria. If you remember this H separately, so that you can remember this disease easily. So other porphyrias are important, but this is much more important than other porphyrias. So you have to remember this. So you have to remember the mnemonic PUC. You have not even completed PUC, Mr. PP. Okay. So H you have to place it here. H we are placing here because if H is not formed, then the disease is known as acute intermittent porphyria. Okay. So the enzyme which converts porphobilinogen to hydroxymethylbilin here, that is PBG deaminase. So PBG deaminase is an enzyme which is essential for the formation of hydroxymethylbilin. So absence of this enzyme leads to a disease called as acute intermittent porphyria. Then other enzymes are a little bit not that much important, but you have to remember. So with the mnemonic PUC, Mr. PP. So U is uroporphobilinogen, then coproporphobilinogen, then protoporphyrinogen, then protoporphyrin. So the last step, using the enzyme ferrochelatase, it leads to the formation of heme. So heme, which is used for the development of RBCs. Okay, so this ferrochelatase, two enzymes which I already mentioned, that is ALA dehydratase and now ferrochelatase. These two enzymes are normally inhibited by lead. So in lead poisoning, these two enzymes are inhibited. So heme formation is not heme is not formed. So here heme is not formed. As I had already mentioned that when heme is a normally a feedback, heme shows a normal feedback inhibition to this ALA synthase enzyme, ALA synthatase enzyme. So ALA is not formed here. When heme itself is not formed in lead poisoning, that is ferrochelatase enzyme is inhibited as well as ALA dehydratase enzyme is inhibited. So the negative feedback from the heme to the ALA synthase is not present. So the negative feedback is not present. So ALA synthase has more action and more ALA is formed. So in lead poisoning, ALA substrate is more formed. So amount of ALA will be increased. And another one, in acute intermittent porphyria, you can find the similar amount of ALA and porphobilinogen which is increased. So both ALA and porphobilinogen is increased in acute intermittent porphyria, and ALA is much more increased in lead poisoning. That is ferrochelatase is inhibited, no heme is formed, so no negative feedback mechanism. So we'll continue like this in other cycles in the next videos. Thank you.